Hello, everyone. My name is Victoria Tuttle. I recently started graduate school at Purdue University, and I'll be pursuing a master's in animal science with an emphasis on physiology. I started working for Dr. Fraley over a year and a half ago, and this talk, as well as the one I did earlier, summarized my projects as an undergraduate student over the past year. And today I will be further investigating the differential effects of glucocorticoids and the sex-dependent pattern on the stress response in pekin ducks. These are the topics I'll be covering today. I will start off by providing some brief background information so that you can further understand my research. I will then go into my hypothesis, talk about the methods, interpret my results, and finally talk about potential future studies I may be interested in doing. So for a long time, people have assumed that the mammalian and avian HPA function similarly, but now we know that this is not the case because there's been a lot of conflicting reports of the stress response in birds, which I will get into in the next few slides. But the main difference between the two axes is the neurohormones involved. Mammals secrete corticotropin-releasing hormone, or CRH, and birds utilize a second neurohormone in addition to this CRH called arginine basotosin, or ABT. So there's been conflicting reports of what the predominant glucocorticoid is in birds. Originally, it was thought to be corticosterone, but results or studies showed a lot of mixed results regarding cortisol levels, and this glucocorticoid release is actually very species dependent. For example, animals such as rats and mice secrete corticosterone, while many mammals, fish, and guinea pigs secrete cortisol, and bottlenose dolphins actually secrete both. So if you were here for my previous presentation, I had a slide similar to this where I compared the two pathways, so I will just briefly summarize. But the neurohormones are highlighted with their locations bolded. The HPA stands for hypothalamic pituitary adrenal axis, but in birds, it's actually more accurate to say the diencephalic pituitary adrenal axis, or the DPA. And this is because the diencephalon encloses the thalamus and the hypothalamus, which contain these avian neurohormones. So HLR stands for heterophil to lymphocyte ratio, and this is an indirect way to measure stress. There is an increase in blood heterophils due to migration from the lymph nodes and a decrease in lymphocytes due to marginalization. It's important to know that the overall number of the heterophils and lymphocytes don't change, just how much is released and the distribution changes in the blood. There's been conflicting reports of the stressors' effects on glucocorticoids and HLR levels, especially regarding transportation stress, which I'll get into detail in the next slide. So studies that looked into transportation stress saw conflicting results regarding corticosterone levels. There's been a lot of variability where studies saw that transportation had no effect on corticosterone levels, while others saw an increase and others saw decreases. And it's not sure if these studies took into consideration the timing of the blood draws and the sex, because we know in mammalian literature that sex differences in glucocorticoid function are due to gonatosteroid-glucocorticoid interactions. And not only do studies in mammals show sex differences, but if you saw my last presentation, these were also present there as well. So Hans Seeley hypothesized the three stages of the physiological response to stress. The first stage is the neurogenic response, and this involves the fight or flight response, as well as the sympathetic nervous system. The second stage is the adaptive response, and this is where the adrenal cortex secretes glucocorticoids to try to adapt to the stressor. And this phase occurs over a period of time beyond the onset of the stressor. As you saw in my previous presentation, the glucocorticoid response to ACTH occurred 1 to 2 hours post-injection, and this was dependent upon sex. The last stage is exhaust or fatigue, and if the animal fails to adapt to the stressor, it leads to deterioration and even death. So I will now talk about some past studies. There was a study done with cockatoos to look at their sex differences in response to exogenous ACTH. The study did not look at cortisol, but there was a similar study done with parrots, and it showed that exogenous ACTH stimulated cortisol levels. Another study was done with mule ducks, and they either force-fed them or gave them an ACTH treatment. And the study saw that corticosteroid levels did not change, but cortisol increased. So cortisol could be a reliable indicator of an acute stressor in these mule ducks. It's also important to mention that these mule ducks are a hybrid between pekin and muscovy ducks, and they are also meat birds. So because they are most likely immature, this points to the possibility of age differences and how ACTH functions in birds. There was another study done with zebra finches to look further into age-related differences. They were exposed to restraint stress, and on the day of hatch, in response to this restraint, they had no glucocorticoid release. But 10 days later, these glucocorticoid levels significantly increased, again showing these age-related differences in glucocorticoid function. Recently, there was a study done, and researchers saw that cortisol was being produced within other organs, such as the spleen and bursa, and they wanted to look further into how immune cell development was affected. They also saw that the bursa in altricial birds has de novo synthesis of cortisol. And these authors showed that this was related to the size and function of the bursa. So we hypothesized that corticosterone and cortisol have differential effects on HLR. The goal of this study was to try to better understand the differential roles of the glucocorticoids. So I utilized adult breeder pekin ducks from Maple Leaf Farms. They are between 35 to 40 weeks of age, with hens and drakes weighing approximately 4 to 4.5 kilograms respectively. They were injected IM with 2 milligrams per kilogram of each glucocorticoid and 0.5 milligrams per kilogram for the low dose cortisol, and safflower oil was the vehicle. The reason why we chose to use safflower oil was because it contains no measurable levels of genistein, which is a plant estrogen. So we didn't want to cause any estrogenic effects on the physiologic response, and that is why we use this as our control. It's also important to mention that we injected equal molar doses of both glucocorticoids, but endogenous cortisol is only a quarter the level of corticosterone, so that's why we added this additional low-dose cortisol treatment to make comparisons. We collected blood at 4 different time points at every hour, and we collected blood smears to look at HLR values, which were then determined in an independent lab with avian pathologists unaware of our treatment groups. So the ELISA kits that I utilized were from Cayman Chemicals, and these were verified previously by Dr. Fraley and his lab. I was not here for this verification process, so I will just briefly summarize what happened. Activated charcoal was used to strip the duck serum to remove any steroids and spiking back known concentrations and comparing to the kit standard curves. Mass spectrometry was also used to validate this process, and this revealed no measurable levels of cross-reactivity between the two kits. So now getting into my results, the graphs are separated to make it easier to visualize, as well as to note the sex differences. There were significant increases in HLR with all of the treatments, and the low-dose cortisol had no effect in the drake, but there was an effect in hens. And if you look at the results for the drakes at the low-dose cortisol, represented by the purple line, there seems to be a trend towards an increase at the 3rd hour mark. So perhaps there would have been a significant increase if we had sampled for a couple of more hours. So if you're here for my previous presentation, I had these top 2 graphs talking about the HLR results, and the bottom 2 graphs are the ones I just went over, and I put these in so that you can make comparisons with the HLR response. So to summarize my results, there were significant sex by treatment by time effects with significant interactions for both glucocorticoids, both glucocorticoids had significant increases in HLR at 1 hour after the injection compared to the controls for the hens. And for the drakes, both glucocorticoids had significant increases in HLR at 2 hours after injection. And finally, the low-dose cortisol had no significant effect on HLR for the drakes, but there was a trend to increase at the 3rd hour. And there was a small significant increase in HLR for the hens at 2 hours. So because we saw the sex differences, this strongly suggests possible age differences because we know in mammals that there are these estrogen receptor, glucocorticoid receptor interactions. Mammals also have corticosterone and cortisol-specific receptors, as well as cytosolic and membrane-bound receptors. There was a recent egg study that was done and found that cortisol was deposited into eggs, but not corticosterone. So with all these results and information adding up from all these studies, we are now hypothesizing that cortisol has a physiological role. And it's important to mention these estrogen receptor, glucocorticoid receptor interactions because this hasn't been investigated in birds. And we know in mammalian literature that the most profound of sex differences in glucocorticoid function are due to gonadal steroid glucocorticoid receptor interactions. So some future studies I'd be interested in looking at is effects of chronic glucocorticoids on HLR and brain function, as well as immune organ atrophy and the deposition of glucocorticoids into eggs, what stimulates AVT versus CRH, and looking further into their differential effects, extraadrenal synthesis of the glucocorticoids, mapping out ACTH receptors and see if they are located beyond the adrenal cortex, and age-related effects on HPA function. So I would like to thank Dr. Fraley for allowing me to be involved in this research. Thank you to my lab partners, Maple Leaf Farms for supplying the ducks, Antech Diagnostics, Finley Research Center, Purdue, and the USDA. Thank you for your time. I will be available afterwards to answer any questions that you may have about my presentation.